Hello everyone. Welcome to my YouTube channel Chemistry Quest. This is the 10th lecture in the series of biomolecules chapter. In this video we will be discussing protein molecules. So let's get started. Proteins are polypeptides which have more than 100 amino acid residues having high molecular mass 10,000 U. U is called unified mass. So there is a distinction between polypeptide and a protein is not very sharp. Polypeptides with fewer amino acids are likely to be called proteins if they ordinarily have a well-defined conformation of protein, such as insulin, which contains 51 amino acid. Amino acids we have already discussed in the previous video. Next, we will discuss different types of protein based on molecular shape. There are two types of protein based on molecular shape. First is the fibrous protein and second is globular protein. First is fibrous protein. When the polypeptide chain run parallel and are held together by hydrogen and disulfide bonds, then fiber-like structure is formed. Such proteins are generally insoluble in water. Some common examples are keratin, present in hair and wool, silk, myosin, present in muscles. These are about fibrous protein. Second is the globular protein. Globular protein results when the chain of polypeptide coil around a given spherical shape. They are usually soluble in water. Insulin and albumins are the common example of globular protein. Next, we will study different types of protein based on their shape. So, structure and shape of proteins can be studied at four different levels: primary, secondary, tertiary, and quaternary. Each level being more complex than the previous one. So, first we will study primary structure of protein. Proteins may have a one or more polypeptide chain. Each polypeptide in a protein has amino acids linked with each other in a specific sequence, and it is this sequence of amino acid that this is said to be the primary structure of protein. Any change in this primary structure, that is the sequence of amino acids, creates a different protein. The simplest structure of protein is the primary structure of protein. Next, we will study the secondary structure of proteins. So, secondary structure refers to the shape in which long polypeptide chain can exist. They are found to exist in two different types of structures. First is alpha helix and second is beta pleated sheet structure. So these structures arise due to regular folding of the backbone of the polypeptide chain due to hydrogen bonding between NH groups of the polypeptide bond. Let's see what is alpha helix. So alpha helix is one of the most common ways in which a polypeptide chain forms all possible hydrogen bonds by twisting into a right-handed circular helix with the NH group of each amino acid residue hydrogen bonded to the CO of an adjacent turn of the helix as shown in the figure. I will explain you this. This is the CO of one helix and this is the NH of one helix. So O and H forms a hydrogen bond. So this is known as alpha helix. Next we will study what is beta plated sheets. In beta plated sheet structure, all peptide chains are stretched out to nearly maximum extension and then land side by side, which are held together by intermolecular bonds, which are hydrogen bonds. So these structure represents pleated folds of drapery and therefore it is known as beta plated sheet. So here there are all peptide chains. This is R, CO and this is NH. So these bonds form hydrogen bonds in between the two sheets. So it forms the pleated folds of drapery. Therefore, it is known as beta pleated sheets. Next, we will study about tertiary structure of protein and quaternary structure of protein. So, in tertiary structure of protein, it represents overall folding of the polypeptide chain, that is further folding of the secondary structure. So, it gives rise to two major molecular shapes, that is fibrous and globular. The main forces which stabilize the secondary and tertiary structures of proteins are hydrogen bonds, disulfide linkages, van der Waals, and electrostatic force of attraction. Next is quaternary structure of proteins. Some of the proteins are composed of two or more polypeptide chains referred to as subunits. The spatial arrangement of these subunits with respect to each other is known as quaternary structure. This is the most complex structure. Let's see this structure in a diagram. So these balls, this is yellow, this is blue, this is pink, this is blue, this is yellow. So this is a simple chain connected of amino acids. So it is a primary structure. Now when it just rounds around some alpha helix, it is a secondary structure. When it forms more coiling, it forms a tertiary structure. And when it forms more coiling, it is known as quaternary structure, where it is difficult to identify the bonds. We will see it is in more simplified way. So in primary structure, this is blue one is the carbon and dark blue is the hydrogen. Secondary structure, N and O are there. N is this red and O is this white. Tertiary structure is little complicated. R is group and heme group is red. So it is coiled around a structure. Quaternary structure is more complicated. Next, we will study about the denaturation of proteins. So what happens is proteins, they are found in biological system with unique three-dimensional structure and biological activity. It is called a native protein. When a protein in its native form is subjected to physical change like change in temperature or chemical change like change in pH, the hydrogen bonds are disturbed. Due to this, globules unfold and helix got uncoiled and protein loses its biological activity. This is called denaturation of protein. During denaturation, secondary and tertiary structures are destroyed, but primary structure remains intact. The coagulation of egg white on boiling is a common example of denaturation. This is the one example. And another example is curdling of milk, which is caused due to the formation of lactic acid by the bacteria present in the milk. So this is all about proteins. If you like the video, please hit the like button, share with your friends, subscribe to my channel, and don't forget to miss the notification bell. Thank you and have a good day.